Real-time monitoring of human guanine deaminase activity by an emissive guanine analog. Guanine deaminase, or GDA, deaminates guanine to xanthine. Despite its recent implications in traumatic brain injury, memory dysfunction, and psychiatric diseases, the study of GDA remains limited compared to other metabolic deaminases. As a result, its substrate and inhibitor repertoire are limited, and real-time activity, inhibitory, and discovery assays are missing. Here, we explore two emissive heterocyclic cores, a thienopyrimidine and an isothiazole pyrimidine, as surrogate GDA substrates, as highlighted by Figure 1. The nucleobases of interest containing either a thienopyrimidine core or an isothiazole pyrimidine core were prepared according to previously established conditions as shown in schemes 1, 2, and 3. To assess the likelihood of distinguishing between the substrates and their corresponding counterparts, the fundamental spectroscopic properties were determined. They are presented in Figure 2. Each nucleobase analog showed unique absorption spectra in water and displayed red-shifted maxima compared to their native counterparts. Molar absorptivity, emissive maxima, and quantum yields for each nucleobase analog are also listed in Table 1. Additionally, their sensitivity to environmental polarity was evaluated, and spectroscopically derived pKa values were determined. To compare the susceptibility of the thienoguanine analog, THGN, and the isothiazole guanine analog, TZGN, to GDA-mediated deamination, enzymatic reactions were analyzed by HPLC and then monitored by absorption and emission spectroscopy. HPLC analysis confirmed the complete transformation of guanine to xanthine and TZGN to TZXN with half-life values of 27 seconds and 21 seconds respectively. The reaction of THGN showed no conversion of substrate. Rewardingly, the spectroscopic analyses agree with the chromatographic analysis. When monitoring the enzymatic deamination of guanine, a half-life value of 31 seconds was calculated. For TZGN, half-life values of 27 seconds and 29 seconds were calculated from absorption and emission measurements respectively. Results are shown in Figure 3 and Table 2. The change in emission signal intensity upon GDA-mediated deamination of TZGN to TZXN facilitates real-time monitoring of the enzymatic reaction and its inhibition. 3 inhibitors were tested, one of them being previously established AICA and 2 new inhibitors. Percent inhibition plots were fitted with sigmoidal hill curves to obtain IC50 values as shown in Figure 4. In this study, we set out to assess the utility of THGN and TZGN as substrates for the enzymatic reaction of GDA. We demonstrate that unlike the thieno analog, THGN, the isothiazole guanine surrogate, TZGN, does undergo effective enzymatic deamination by GDA, and yield the spectroscopically distinct xanthine analog, TZXN. The presence of the nitrogen equivalent to the N7 in the purine skeleton endows TZGN with the substrate recognition features necessary for the reaction. This discovery provides a biochemical tool to study the activity of human GDA. We also illustrated the potential through a small pilot study of inhibitor effects on GDA activity.